maniaking way. Okay, our last aspect of proteins, and this is protein folding. We recall that protein's function is dependent on them folding up into a proper tertiary structure. But that process is slow for most proteins. That's why if you denature a protein, the vast majority of them will clump together by their hydrophobic exposed regions far faster than they can fold back up again. Clumping may take fractions of a second. Folding can take minutes in some cases. It's a very complex thing. And it's something that all people are trying. It's something we cannot do by a computer model or anything. Not yet. People are working on that, but it's just a very difficult problem. Okay, well, now think of this in a cell though. You're going to have proteins at very high concentrations. Cells are about 20% protein by weight. That's a heck of a lot of protein. Concentration, 200 milligrams per milliliter. You can't, it's almost impossible to take a purified protein and make a solution that concentrated. So there's a huge amount of proteins in cells. Now sometimes proteins are going to get denatured. Question is, if a protein gets denatured, is there a chance it can be salvaged? Maybe. Think of this. When you're synthesizing a protein, you're going to have a messenger RNA, you're going to have a whole pile of ribosomes lined up on that. Each ribosome is going to be reading that messenger RNA and synthesizing a protein. So now a protein starts coming out and coming out as we continue down the messenger RNA. Now, those proteins have to fold up. And the problem is, these newly synthesized, unfolded proteins are so close to each other that they swing around a little, their hydrophobic parts can stick, and you get a bunch of gloppy, clumped together mess, unless you have something to do about that. So that's another issue. So far we got a situation. Can we sometimes rescue denatured proteins, help them to fold back up again before they clump? In a test tube, forget it. In cells, the answer is sometimes. Okay, newly synthesized proteins. We got to prevent these guys from clumping together, and we've got to help them to fold up. Give them a little bit of time so that they can fold up properly. Okay, third thing we have to do. Sometimes we have to get proteins across cell membranes. Now, protein by itself is not going to be able to get across the cell membrane on its own. It needs help. And help comes in the form of certain protein channels. The problem is those channels have a pore too small to fit a fully folded protein through, just like that top lid on the proteasome. So in order to do that, what you have to do is you have to be able to take that protein, unfold it, stick the unfolded protein through the channel so it can fit, and then the other side of the membrane, you have to fold it back up again. So you have to take it, unfold it, stuff it through the channel, and refold it at the other end. That's how you get things across membranes. Now the question is, here's 3 biological problems we got to solve. How do we do it? And that brings up the group of proteins that do these things. And these are called the chaperones. They're a class, in fact, they're a couple classes of proteins. And what they are, proteins are designed to help other proteins fold up, protect them from clumping, or in some cases, actually deliberately unfold and later refold the protein. And in our genome, there's genes for about 400 different chaperones, although they do fall in about 3 or 4 major categories. We also find chaperones in prokaryotes, bacteria and archaeans, although they are different from the ones we have. They have some similarities but some differences as well. These chaperones are all over the place, and once again, they are essential for life. Get rid of a chaperone gene, and that probably means you're dead. And these are the proteins, and we'll see how in a minute, that carry out these various functions. Okay, now a little bit of history behind this. Just a little historical interlude. Way back in the 1960s, people realized when they were growing cells in culture, or things like fruit fly embryos in culture and stuff like that, they noticed that these cells, or in some cases embryos, got exposed to slightly elevated temperatures. Instead of like 37 body temperature to about 40, 42 or so, hot summer day in Alabama kind of temperature, they noticed that what happened is these cells would basically stop producing just about all kinds of proteins and instead produce huge amounts of one particular protein. And then when things cooled down, they would stop producing this protein and start producing everything else. So they noticed that that was a common property of cells. So because it seems that cells exposed to excess temperatures could cause sudden massive production of these proteins, they started calling these things heat shock proteins. And then later they found out that elevated temperatures weren't the only things that could cause huge productions of these proteins at the expense of all the others. Instead, they found if they exposed cells to other kinds of stressful things, sometimes cold, organic solvents, detergents, things like that, that could also cause the same thing. So they started calling them stress proteins. Stress out the cell, and you get large amounts of these things produced. Once they had the techniques of genetic engineering, they found that if you got rid of the genes for these, which identified for these heat shock proteins, and then exposed cells to nasty conditions and stuff, the cells died. They could not tolerate even slightly elevated temperatures if they didn't have these heat shock genes present. Likewise, any other kind of stressful thing. And people start realizing, what do these things, all these nasty stressful things have in common? They denature proteins. So maybe there's a connection between these heat shock or stress proteins and protein denaturation. Is it possible that these proteins are a damage control team to try to rescue and refold denatured proteins? Then later on they found that there were other members that were very similar that were present all the time. So we had these proteins, and that's when they started coming up with the term chaperones and stuff. There are certain kinds of chaperones that are always present, what we call constitutive. That means it's always there. And they found there were these constitutive, these always present heat shock or stress proteins, chaperones. And then they found kinds that were what we call inducible. In other words, they're normally not produced, but they are produced when we need them. Those are the classic heat shock proteins. So the inducible versions are the cell's damage control team. All of a sudden, the cell realized, oh my God, nasty, horrible conditions, proteins are getting denatured left and right. Call out the rescue squad. Call out the cellular SERPRO like it never even happened. Okay. All right, what happens? All of a sudden you get all these all these chaperones, and they try to fix up the damage. Now obviously there's going to be limits. You can overwhelm them, so cells will still die if you heat them up too much, but it gives you additional resistance. But we also have the ones that are always present, playing all kinds of different important roles in cells. And some of those roles include helping to prevent newly synthesized proteins from clumping together and gluing together before they can fold up properly. Rescuing denatured proteins. They've got their limits, but they can help. Helping proteins to fold. Now, how does that work? Well, despite all the different types of chaperones, and there's variations on this thing, they have certain common mechanisms. One of them is that all the chaperones are ATP binding proteins. They bind ATP. They're also all ATPases, which mean they hydrolyze ATP, toss out the phosphate, now they have ADP. And then they can toss out the ADP and pick up an ATP. Now the neat thing about this is they have two structural states. And depending on the chaperone, which one is which varies, but okay, one of the structural states that chaperones have actually has a bunch of really, really hydrophobic amino acids on the surface. What we sometimes call a greasy spot. And that spot is loaded with hydrophobic amino acids. Needless to say, it does not want to be in water. It wants to be in a hydrophobic environment. Now the other structural state of chaperone, that hydrophobic patch is buried in the interior of the molecule, so it's no longer on the surface. Depending on the chaperone, sometimes that's the ATP bound state, sometimes that's the ADP bound state. It varies. But that illustrates the way that chaperones work. So we've got two structural states that are dependent on ATP or ADP. One's got that greasy spot, the other doesn't. Now let's suppose we've got ourselves a protein here. It's not folded up properly, and the squiggly line represents the hydrophobic stuff. Now once the protein properly folds, that hydrophobic stuff's going to be, for the most part, in the interior of the protein. But while it's still exposed, it can grab on and clump together with some other unfolded, denatured, or not yet folded protein. This is where the chaperones come to the rescue. What they'll do is that real greasy hydrophobic surface patch will stick to the hydrophobic parts of that protein. It's going to protect it. It's actually a little kind of furrow in the protein. They have tertiary structure information on a few of the types. It's a hydrophobic furrow. So here, this nice hydrophobic section of amino acids fits nice into the canyon, and that means nothing else can get at it. So it's bound to the shell. Okay, now, while that's going on, it's possible that the protein will start to fold up. So we get partial folding. Now, the chaperone hydrolyzes ATP, or whatever the case, and now we get rid of that hydrophobic patch, and the protein is released. By that time, the protein may have partially folded. If you're real lucky, it's folded completely up. Okay, so we release the protein. Now we go through an ATP-ADP cycle again, and now the chaperone has that hydrophobic patch. If the protein is fully folded up like so, it's not going to bind to the chaperone anymore. But let's suppose, like this example, it's partially folded. You still have exposed hydrophobic portion. So it sticks to the chaperone again, and the chaperone gives a little bit more time to refold. And then it releases it again. And maybe the protein folds up a little bit more. Once again, we go through the ATP cycle. If there's exposed hydrophobic regions, the chaperone can rebind to the protein, give it a little bit more time to fold up, and it keeps on binding, giving it time to fold, releasing the protein, rebinding, and finally back and forth until the protein is so folded up that now it's no longer capable of binding to that hydrophobic patch. So now we have a fully folded protein, and it will not stick anymore. So chaperone has done its job. So what chaperones do, especially these so-called constituent, the ones that are always on, always there, what they do, things like newly synthesized proteins, they protect them from clumping with other proteins, and they give them a shot to fold up. In fact, they give them a lot of shots. If you have denatured proteins already, especially the rescue squad and the heat shock proteins, those guys will help to allow at least some of them to fold up. Now, some chaperones can work in reverse. They can bind a protein and actually denature it. And those are the ones that are going to be involved in getting proteins across cell membranes. Yes?
What is the last word of the first part? Region from?
From clumping.
Clumping.
Yep. Okay. So now we've got that. So this is a very important kind of thing, allowing proteins to fold up. Now an interesting thing, despite, especially newly synthesized proteins. Now an interesting thing, the chaperones are not God, okay? They can help, but they are not a guarantee. So they will not be able to rescue all denatured proteins. If that happens, the ones that still haven't folded up properly get tagged with ubiquitin and destroyed. When you get proteins across the membrane, you have to unfold them, slip them through the channel across the membrane, and the chaperone at the other side of the membrane tries to fold it back up again. Maybe half of them don't fold up. Guess what? They get pushed out of across the membrane again, tagged with ubiquitin, and destroyed. Perhaps half of our newly synthesized proteins clump rather than fold. End result, tagged with ubiquitin and off to the proteasome. So, inside the cell, the cardinal rule is for proteins is fold or die. Failure to fold is a capital offense inside proteins, and ubiquitin, judge, jury, and the proteasome executioner are always waiting for another victim. But chaperones help out. They are absolutely essential for life, folks. And all cells from prokaryotes on up to us have numerous different chaperones of different types. And that covers our investigation into the world of proteins.